Thank you, Roberta, for introducing us. I did not get a headset, and you spoke Italian, so I'm not sure I understood what you said. But anyway, thank you. Thank you to Enoforum for inviting us both to take the floor this morning. We talk about non-conventional yeasts, non-Saccharomyces yeasts. One specific application of these yeasts pertains to base wine for sparkling wines. So we decided we would see whether we can work on these yeasts and apply that to the other wines as well. I will talk about Hanseniaspora vineae only. I'm not a microbiologist. I will not dwell upon technical details about microbiology. Anyway, this is a yeast that has already been mentioned. H. vineae is to be found on the skin of grapes. It has this sort of lemon-like citriform. It is featuring bipolar budding. Ethical resistance is low, which is the reason why oftentimes Saccharomyces is added. And I repeat, I'm not a microbiologist, but I know that it does not use NH4. At least I think, since I'm not a macrobiologist. There is then another special aspect to be considered, which is that it produces very interesting aromas, as is the case with the phenylethyl acetate. Let's have a look at a number of details. Antonio Morata's group worked on this topic, and then further groups focused on this in Australia. Most studies concentrate on the sensory impact of Hanseniaspora vineae on its own or together with additional yeasts. So on the top of the slide, you can see as an author, Martin Valentina, who focused on the capability of this yeast to create new aromas thanks to the impact on cell lysis. And this is when it is in contact with lees. Then and this is something that Antonio Morata already mentioned. Here you see aging on lees and how it develops. It is a rapid process when it comes to this yeast. There has been a comparison with the situation when adding Hanseniaspora vineae and the excretion of biopolymers. A comparison is made with the two Saccharomyces and other yeasts. So theoretically, Hanseniaspora vineae releases proteins and also polysaccharides. This is a special characteristic, which is aromatic and which is interesting. As I said, it can also be used and exploited in sparkling wines, especially with reference to the creation of foam. Oftentimes, I was told that we should work on that because it is ephemeral, very difficult to understand. What does this picture tell us? Well, you see, this foam formation is a rapid phenomenon and then it disappears again. MacLeod, more than 20 years ago, stated the information processed most quickly by the brain and which reaches consciousness first is visual information, when other sensory representations provided by the tongue, touch, or hearing reach the consciousness, they confirm or contradict the expectations induced by visual information. So visual information is important. The other sensory information linked to taste and smell can confirm or not the first impression which comes from sight, which means that bubbles and foam are key, the first criterion for the quality of sparkling wines. That goes for all beverages and also sparkling wines. Let's briefly comment this slide. Bubbles and foam are little explored. My colleague will talk about the details about this. You see, there is an important property related to that, which is the release of biopolymers that can contribute to the volume you feel on the palate. Olivier will focus specifically on that. Foam. Here you have two beverages. You have sparkling wine and beer. On the left-hand side, you have many bubbles, very little foam. On the other side, you have beer, very few bubbles and a lot of foam. This is paradoxical as a behavior. We need to find a way to explain that. Alcohol does not help stabilize the foam, and that is the reason why the figures are in red in the sparkling wine column, while it stabilizes foam. So in beer. So what you see in green is what explains the different behavior of sparkling wine and of alcohol. Since sparkling wine has a certain content of grams of proteins, milligrams in champagne, maximum 100, and in beer there can be as much as 1 gram of protein per liter, which has a stabilization effect on foam in beer. Then polysaccharides are another very important biopolymer. There are as many as 10 grams in a number of beers, while the quantity is different in sparkling wines. So let's focus on the importance of biopolymers on foam. We extracted glycoproteins from wine, and we added these proteins to a base wine, which did not contain biopolymers. When you add 10 milligrams of this mix of glycoproteins to a base wine, which otherwise contains none, you can create a bubble. We used a laser a laser device to see the bubbles, and they explode at a certain point because they are not stable. If you add 30, 40 milligrams more of glycoproteins, you see that there is more stability in the bubble. It lasts for much longer. And the color is also different in that light passes through, and the thickness of the film is so thin that it is just in the order of nanometers. Little concentration can stabilize the foam thanks to this mix of biopolymers, glycoproteins, mannoproteins, just to mention one group. The mechanism is relatively well known. but I would like to tell you about this because it is important. It explains the stability of the bubbles, and it also tells about the behavior of foam in beer and in some wines. Certainly, there is a drainage effect of the liquid, which is the film of the bubble. So proteins are capable, by means of electrostatic repulsion, to attack the interfaces. If there are glycoproteins, mannoproteins, then there is a steric hindrance, so much so that the interface is not there, which means that a limited quantity of matter can already stabilize foam. Let's now go back to aging on lees. You know that there is a release of mannoproteins in a considerable amount. And foam stability and complexity of the wine are related to that. The yeast autolysis process is relatively slow. You realize that this has taken place after 4 months. Maximum is reached after 2, 3, 4 years even. And we see here an example of polysaccharides on the left-hand side and proteins on the right-hand side. And you see the autolysis quantities in red for yeasts. So after 4 years, everything is completed for both polysaccharides and proteins, which means that this is a process which lasts for months, if not years. If you remember what I showed to you in the beginning, you see very well that we can have interesting applications for Hanseniaspora vineae. This is a very interesting work by Alexandre and Guillaume Benatier about the cell content and cell wall of yeasts supporting foam, and also all the other things that have to do with the volume as a mouthfeel and the texture of wines. Now, we have a yeast that we would like to use for non-sparkling wine. We have grape must, which can be added with Hanseniaspora vineae, and we focus our interest on finished wine with bubbles after a long winemaking process. That process is, as I said, long and also more invasive. There is a property of the yeast to release biopolymers, which is used during the pre-demousse, and coadjuvants are used, especially in the remuage phase. Bentonite is what I refer to. That is the function of bentonite, which is that of trapping biopolymers which have been excreted by the yeast. We could now see to what extent that happens. We do have an answer about that. Again, we have several groups tackling the matter in Italy, in Australia, in Spain. Here on the left-hand side, you see bentonite. We're not interested in knowing whether it has been activated or not. But you see that low molecular weights are specifically impacted by bentonite. And there are glycoproteins, mannoproteins. This is still wine. You see on the right-hand side, cava wines, pre-demousse. When there is no bentonite, there is a huge difference for medium and low weight, molecular weights. Consider this fraction. with a low molecular weight that has been spared by bentonite, which means that we can hope that Hanseniaspora vineae, despite the fact of being still remote from application in the wine, could help improve the quality of sparkling wine, and I'm referring specifically to texture and foam. Should we remember something to take home as a message before giving the floor to Olivier, who will try and explain to you the results that have been obtained with Hanseniaspora vineae and alcohol fermentation, I would like to tell you the following. Bubbles and foam are the first quality criteria for sparkling wine, so that should not be forgotten. Glycoproteins and proteins have an impact on the mouthfeel and help stabilizing foam. Then yeast autolysis is important because it is stabilizing bubbles, and yet it is a slow process. Plus, Hanseniaspora vineae during fermentation, malolactic fermentation, can release the biopolymers and therefore can possibly be an alternative to autolysis. This is still an open question. Olivier will focus on that and will try and explain this to you based on the results of the study. But before giving him the floor, I would like to present a number of methods. You remember the pictures of the individual bubble. That is a method where you focus on the individual bubble to quantify foam. There are also other methods whereby you observe three-dimensional foams, and I think that Moselux is famous, known to you all. It is called Rudin foam something in English. You see how rapid this test is. Foam is hard to manage, difficult to manage, so repeatability is not something that can be easily accommodated. Olivier will tell you more about HS, which is a situation in which there is a balance between the foam and the rest. So the same number of bubbles are created and destroyed. And then there is the soap maker foam test, which permits to make interesting observations in terms of numbers, and you can repeat the text approximately 20 times over the course of the day, and to measure the development of foam in time, and it is a more repeatable option as a test. Now the floor goes to my colleague, Olivier, who will tell you about the interest of Hanseniaspora vineae for sparkling wines.
Merci beaucoup, Bertrand.
Thanks a lot, Bertrand. Hello, everyone. I'm trying to respect the Italian language. Out of respect for the Italian language, I will speak in French. Now, as Bertrand pointed out, I'll be talking about the yeast. He was talking, and Antonio Morata also discussed it earlier this morning. It's interesting in the spumante and sparkling wine making industry. Now, as Antonio Morata has mentioned this, I think generally the word Hanseniaspora is scary. It's a totally different species. Hanseniaspora vineae is not the same as H. uvarum, which is a family of yeasts that can produce ethyl acetate, which is not the case with Hanseniaspora vineae. And problems like these are much less frequent with H. vineae. Now, we took an interest in quick autolysis that can be produced by H. vineae. We have papers that show the interest of certain strains in view of the release of polysaccharides. As you can see here, in their positioning is positive compared to other yeasts, and this is what we were looking for. What we're interested in is not really the quantitative result, but the qualitative outcome. The fact of having different mannoproteins with different sensory characteristics. In this case, this is what appears to be the outcome of the fermentation carried out and brought about by H. vineae and its mannoproteins. So when you talk about mannoproteins, you also discuss colloidal stability, and what we're also interested in is the stability of the foam. The strategy we used in this approach was about taking an interest in Hanseniaspora vineae and associating it to an Cerevisia. As Bertrand pointed out, H. vineae does not live well with high levels of ethanol. And so we had to carry out several tests on H. vineae together with a strain of S. cerevisia, which is known for its tolerance, ethanol tolerance. There are strains which are known for being contributors to the acetyl-phenyl ethyl, which makes the aromatic profile more complex. We didn't want to push this too far. However, in order to prevent an overpowering floral notes, which are not always desirable in sparkling wines. So complexity, okay, but also preserving the typicity and the profile of the wine. What we wanted to test in our research when coming to this association of yeast was preserving the pure H. vineae strain, and whether this minimization of the acidity by coating it, the expression of roundness and volume, and the stability of the foam first and foremost. This was our strategy. Now, we used H. vineae in a pure state, which is the key element in this mix, and we could confirm that this strain during alcoholic fermentation can release higher amounts of mannoproteins. This is a Chardonnay-based wine, and you can tell there's a significant difference. If you look at the wines that result from a fermentation of a mixture of yeast, where the market name is Divina, you find the same result as with a pure strain. The characteristic of Hanseniaspora vineae is preserved even when it is associated and combined with this higher mannoprotein content that is mixed with the base wine at an early stage. I would like to thank the MAC Foundation for contributing to this research with their vinifications, first pilot lab scale vinifications and then winery scale vinifications. Now, let's discuss the base wine. Bertrand discussed the soap maker's test, which is pretty easy, which we carried out on the wines fermented with H. vineae. The foam is, in fact, more persistent when the mixture of yeast was used versus the pure yeast. This was reproduced with another matrix and also with a different Chardonnay. The result was confirmed. We have a thicker foam and that is more persistent and stable over time. We also worked with this yeast mixture, and the mixer leads to exactly the same consequences in terms of foam thickness and stability versus the Saccharomyces strain, which was the reference for these trials. This is also visual. We measured the thickness. We used the Moser-Lux test, very similar. Bertrand made reference to it. And we have exactly the same results with much higher stability in the foam and a longer-lasting foam compared to fermentation obtained with only S. cerevisiae. Now, Monselux was used in order to analyze the duration, the time of stabilization and duration of the foam. And this combination of yeasts grants more stability thanks to the combination in yeasts. And the difference can be very, very marked and evident. You find better results with the higher thickness of the foam in the tests carried out also on other wines. So these are precious indicators that come from the trials. So when you compare the average between the controls and the experimental sample, then you have significant and interesting results. So when you take an interest in the mix of yeast and non-Saccharomyces yeasts, the fermentation changes a bit. It's a bit more sluggish at the beginning, but actually, there's a good triggering, and the start of fermentation is slightly delayed, but then the speed is reached and the fermentation is completed successfully and very quickly thereafter until the full fermentation of the sugars. And that guarantees the fact that these yeasts can truly ferment properly the sugars. We also assessed on top of the foam persistence the sensory profile. We know there's a variability in which sparkling wines are served, are poured. There's some variability in the tasting, which was carried out in Spain, in this case, with a longer-lasting foam in the samples fermented with a combination of yeasts. A less aggressive sparkliness to the palate, so the bubbles were not so aggressive to the mouth mucose. And so it's a better mouthfeel, more roundness, more sweetness to the palate, which is interesting when you work with this yeast combination. Also, the aromatic intensity was higher, and what we're interested in is actually the aromatic intensity, which was found to be satisfactory in these yeast combinations versus the yeast on its own. Now, let's look at the consequences. In this paper, we wanted to validate the complementarity of Hanseniaspora vineae with its unique sensory properties with a fermentative yeast, a robust fermentative yeast, for the first fermentation of base wines, which was achieved successfully while respecting the aromatic complexity of the wine. These tests were taken care of considering that H. vineae has a quick autolysis capacity, early and powerful release of natural yeast compounds, which in sensory analysis was leading to foam stability and roundness, less acidity, and a reduction in the aggressiveness to the palate. This is important because we're playing around with something that it is very hard to stabilize, foam, and we now have a tool that can help us impact this particular parameter positively. Now, here we have an alternative that allows us to have a natural active ingredients that will improve the perception of roundness and volume to the palate, but without any need to change your label indications because they're not gum arabic nor anything else that would impact the wine negatively from that perspective. Hopefully I was clear enough.